Identifying the specific genetic biomarkers that differentiate acute lymphoblastic leukemia from acute myeloid leukemia allows clinicians to assign the correct therapies. Finding those markers requires isolating a faint biological signal from an ocean of cellular noise. We can observe this noise in the 1999 GOLUP dataset. The raw gene expression data forms a matrix. The y-axis contains 3,051 individual genes, and the x-axis contains 38 patient samples. The group structure is encoded into a binary variable. 27 samples represent ALL patients, and 11 represent AML. This matrix illustrates the high P, low N statistical bottleneck common in genomics. We have an immense number of variables, the genes, but a limited number of subjects. Extracting a reliable expression pattern from this dimensional imbalance requires advanced statistical machinery. Without it, the sheer volume of data guarantees that mathematical artifacts will be misidentified as actual biomarkers. If we only wanted to evaluate a single gene's expression between the ALL and AML groups, a standard hypothesis test, like a t-test, works. It calculates whether the difference between the two groups is statistically significant. But applying that single test to thousands of genes simultaneously causes a statistical breakdown. As the test fires off across all 3,051 rows, probability dictates an accelerating number of false positives. This creates a high false discovery rate. The FDR is the probability that a significant result is actually a mathematical illusion. To safely explore this dataset, we must control the FDR. We will do this by running the numbers through two distinct statistical engines, significance analysis of microarrays, SAM, and lemma. Without rigorous FDR control, evaluating thousands of simultaneous standard p-values leads to unreliable biological conclusions. SAM bypasses complex probability formulas by relying on a brute force computing architecture called resampling. To establish a baseline of pure random chance, SAM permutes the dependent variable. It detaches the labels and shuffles them across the data. This destroys the real biological signal, calculating a meaningless test statistic. Iterating this thousands of times builds an expected null distribution. SAM compares genes exceeding a threshold in the real data against the average number exceeding that threshold in the permuted data. This comparison allows us to calculate how many of our findings are likely to be false discoveries. SAM utilizes computing power to mechanically construct the baseline of random chance, circumventing the need for theoretical assumptions. To separate the biomarkers from the noise, we define a parameter called delta. Delta establishes a mathematical boundary for statistical significance. This scatterplot displays expected SAM values against observed SAM values. Selecting a delta of 1.11 locks the false discovery rate at 0.05. Genes outside this threshold are flagged. A secondary 2.0-fold change requirement ensures the expression difference is at least a 100% deviation. Filtering the original 3,051 variables through these parameters leaves a final output of 88 upregulated and 43 downregulated genes. SAM isolates biomarkers by combining mathematical FDR control with physical biological magnitude. Lemma approaches the exact same dataset using a distinct mathematical framework based on empirical Bayes methods. Before calculating any statistics, the sample group structure is encoded into a design matrix. This algebraic layout assigns binary zeros and ones to align the samples into their ALL or AML categories. Lemma then utilizes the LM-fit function to fit a separate linear model for every single gene across these predefined groups. Next, the make contrasts function is applied. By setting the specific contrast of ALL minus AML, the software isolates the precise vector of expression difference between the two diseases. Before Lemma can apply its variance adjustments, it requires absolute algebraic precision to define which biological groups are being compared. In our dataset, the AML group only contains 11 samples. Calculating a standard deviation from such a small sample pool results in extreme volatility. In the standard t-test equation, a volatile variance in the denominator destabilizes the entire calculation, making the results mathematically untrustworthy. Lemma uses the e-Bayes function, applying empirical Bayes methodology to borrow strength. Rather than relying solely on the 11 AML samples, Lemma draws a global variance prior from the entire matrix. Information from the other 3,000 genes stabilizes the individual gene variances. This newly stabilized equation produces what is called the moderated t-statistic. Using the top table function, the pipeline applies a Benjamini-Hochberg FDR correction and filters the data for a log2 fold change of at least 1. Lemma's final output isolates 129 significant genes. By leveraging the behavior of the entire dataset to mathematically backstop each individual gene, empirical Bayes methodology rescues the t-test from the high-dimensional environment. This volcano plot visualizes Lemma's outputs. The x-axis tracks log-fold change, the y-axis tracks statistical significance. Points pushed high and wide are the strongest biomarker candidates. Threshold lines drop in at plus and minus 1, cornering significant genes into the top quadrants. Comparing Lemma's 129 extracted genes against the 131 genes isolated by SAM verifies our results. The overlap reveals near-perfect alignment. The intersection contains 127 identical genes. There are only 2 outliers selected by SAM, but not by Lemma. One pipeline utilized a permutation-based resampling approach. The other relied on Bayesian probability and linear models. Despite these different mathematical paths, we find a consensus on which genes are the most likely biomarker candidates. We can now move from analyzing abstract statistical variables to visualizing the actual expression patterns across our specific patient samples. By mathematically centering the expression values of these specific genes against their mean, we make the patient profiles directly comparable. This hierarchical clustering heat map provides the predictive model. The centered expression values display in red and blue. Each row represents one of our curated genes, and each column represents a patient. The dendrogram branching across the top shows that based entirely on the 129 gene profile, the algorithm correctly clustered the clinical groups, splitting the ALL patients from the AML patients. A single ALL patient clusters on the opposite side. This outlier serves as a reminder of the limits of physical data and the reality of natural biological variance. These methods reduce an impenetrable wall of data into a specific biomarker profile, identifying the genes that define the difference between these two diseases.